Hello and welcome to another MCQ practice session. Today's topic is MolBio and this is the session number 3. If you want to access the previous session, you can click on the I button to get the entire playlist, and it is distributed topic wise. There will be 10 questions and we'll solve it in next 10 minutes. Now, if you don't know what to do in the last 2 months before IIT JAM 2022, then this kind of video is exactly for you. In this last 2 months, there should be a balance between MCQ practice and conceptual revisions. And in last months, tricks, tips, and strategizing your examination can really help you to get good rank. So from that perspective, Unacademy's new ambition batch could be really beneficial for you, which is starting from 15th December. And you can obviously take a subscription for this batch by clicking on the link which is provided in the description. If you use my code AP10, you will get an instant 10% discount. Now let me tell you the benefit of these batch courses. First of all, top educators throughout India would take your class and help to clear your doubts during these critical time. There would be daily mock tests, there would be weekly test evaluations, there will be doubt clearing sessions which would be really helpful for your last moment preparation. Now, right now, due to Christmas, there is another offer. If you take a 12 months or 24 month subscription, that would give you 2 months or 4 months extra. Now, if you are a second year or first year student, this kind of particular offer is really important for you guys and really beneficial for you guys. So don't forget to check out the link in the description. If you plan to join right now, because this is the most beneficial time to join. Anyway, let's move on to question number 1. So this question says, which of the sequences listed below best describe the order in which following enzymes participate in replication of DNA in bacteria? It says primase, helicase, DNA pol 3, DNA ligase, and you have to order them in a particular sequence of their activity. So obviously here the correct answer would be option A, because helicase acts the first and it unwounds the strands. Then there would be then there would be primase, which form the primer. Then there would be DNA polymerase 3, DNA ligase eventually will seal the gaps. So that is why option A is correct. Now let me tell you that these kind of questions are very common in IIT JAM, where they ask about like several enzymes used in central dogma processes like replication, transcription, translation, etc. So you can have a list of these. Which of the following reaction is required for proofreading? That is correcting replication errors during DNA replication by DNA polymerase 3, right? So it simply tells us that which of these following activity is responsible for the proofreading activity. And the correct answer would be 3' to 5' exonuclease activity. Now, in order to know how DNA polymerase work, you can click on the link which is provided in the I button. You would quickly revise this concept within 5 minutes. Question number 3. T4 DNA ligase requires ATP, joins double-stranded DNA fragments with an adjacent 3' phosphate and 5' hydroxyl, requires NADH, joins single-stranded DNA. Now, the correct answer here is it requires ATP. And if you need the details of how ligase enzyme work, you can always watch the video on I button. Question number 4. Mitochondrial DNA replication mitochondrial DNA is replicated from a single ORI site bidirectionally, 2 different ORI sites, many sites bidirectionally. Replication does not require any ORI. And the correct answer here would be option B, which requires 2 ORI sites. Question number 5. All the following factors are required to initiate translation of mRNA in prokaryotes except formyl methionine tRNA, 30S ribosomal subunit, guanosine triphosphate, elongation factor TU, EFTU. The correct answer is EFTU because it's an elongation factor and it's not required to initiate the translation process. If you want to revise translation very quickly, which could be confusing to you guys, click on the link in the I button right now. Question number 6. Shine-Dalgarno sequence of mRNA is helpful in recognition of 50S ribosomal subunit, recognition of 60S ribosomal subunit, recognition of 5S, and 30S ribosomal subunit. And the correct answer is the last one, recognition of 30S subunit of the ribosome. The Tata box plays a key role in assembling active transcription complexes by binding with Tata box binding protein, binding with DNA protein, binding with RNA protein. It does not bind to any protein at all. Correct answer is Tata box binds to the Tata box binding protein. So general transcription factors like TF2D family has this Tata box binding protein domain. So that binds with the Tata box and that allows the assembly of the transcription in a transcription initiation complex around the promoter, and this is how transcription happens. You can always cross check that by clicking on the link in the I button. Question number 8, it is from lac operon, and these kind of questions are really common. Cyclic AMP regulates the lac operon by binding to the operator to turn on transcription, binding to the lac repressor to prevent transcription, combining with the catabolite activator protein to form a complex that turns on transcription after binding near the promoter. None of the above. Here the correct answer would be option C. Right? If you want to learn in details, you can find lac operon video in the I button. Question number 9. Which of the following modification can simultaneously take place in DNA as well as histones? Acetylation, methylation, ubiquitinylation, palmitoylation. Correct answer here would be methylation, because methylation can occur in DNA as well as histone. Histone can be methylated, DNA can also be methylated. Now, the last question says, which of the following DNA polymerase in eukaryotes has 5' to 3' exonuclease activity? DNA pol alpha, DNA pol beta, DNA pol gamma, none of these. Now, for this question, I'm not going to tell the solution. You can write the solution in the comment section. And see you in the next video. And as usual, don't forget to like, share, and subscribe. If you have a suggestion, put it in the comment section. Thank you.